Good afternoon for most of you. Good morning for our speaker today, who's coming all the way from California. Well, he's in California. You look at the background picture. Who is a friend of IMO and a colleague for many, many years, is Dr. Rockne from City of Hope, which is another cancer center and NCI designated cancer center from the area of LA. And Russ has been, we have a lot of panelists on the screen now. Russ has been a part of the math oncology community for a long time. He did his PhD in maths at the University of Washington under the supervision of Kristen Swanson, someone else who you guys will recognize. He did a postdoc at Northeastern and started his own group at City of Hope a few years back, where he heads the division of mathematical oncology. So one of these places where you can actually work and do mathematics in a cancer center. So great work. He has been incredibly successful in terms of getting research published and getting grants to support our research. His group is growing, and I'm pretty sure that his position as assistant member and assistant professor will be upgraded very soon to tenure associate professor. He has been working in a number of areas, going from the more fundamental mathematical oncology, the more theoretical aspects, to the more clinically oriented ones as well. So his research covers all areas. And without further ado, Russ, please take it on.
Okay. Thank you, David. That was a nice introduction. Hi, everybody. I thought, first of all, thank you for having me, everyone at IMO, David for the invitation, and Sandy and everybody. This is a presentation and talk I put a lot of thought into, because this is, you all are my people. We're all together here. And so I chose this particular topic because I thought it spoke to a lot of common interests that we have between our two groups, East and West Coast here. So the title is a little bit long, because academics like to do that, but it also tells you what I'm going to talk about, and that is targeted radioimmunotherapy and CAR T-cell combination therapy for multiple myeloma, from the bench to math to bedside and back again. Okay. So because there's a lot of content here and there might be new concepts for people, let me just tell you kind of how I structure the talk. First, I want to tell you a very brief introduction about what multiple myeloma is. And then what I mean by radioimmunotherapy may not be what you instinctively first think it means. And what is CAR T-cell immunotherapy, a lot of people are familiar with, and combination therapies, and why this combination of RIT, I'll call it RIT, radioimmunotherapy, and CAR T-cell therapy is particularly potentially impactful and also challenging. And then mechanisms of resistance, and I hope I get all of your attention with this one, because we have a different take on mechanisms of resistance than I think a lot of the current views are, and hopefully this will also encourage you to pay attention all the way to the end. Okay, but warning, this is a work in progress, so I'm going to show a lot of unpublished data, a lot of things that I haven't, that aren't published yet, so a lot of opportunity for discussion. Okay. Multiple myeloma is a disease of bone marrow and plasma cells. Effectively, in normal healthy bone marrow, you produce plasma cells and antibodies, but in multiple myeloma, this is aberrantly overproduced, both the plasma cells and the antibodies. And it's a fairly rare disease as far as cancers go, and just in general, it's only about 1.8% of all new cancers, 32,000 new cases expected this year. It has a fairly good overall survival rate, 5-year survival rate of about 54%. But it really is a disease of disparities, so it disproportionately affects the Black population, both in incidence and death rate. And I'm not going to talk about that in this talk here, but it's something definitely worth noting. And it's also a disease of aging, like a lot of cancers are, so median age of diagnosis is about 70. Daratumumab is a monoclonal antibody. It goes under the name Darzalex. I'll refer to it as Dara. It's a human antibody against the receptor CD38 and has FDA approval for multiple myeloma patients that relapse. CD38 is a cell surface multifunctional ectoenzyme, so to speak, receptor that is important for regulation of calcium and signal transduction. And CD38 is highly expressed on plasma cells, and it has multiple mechanisms of action to kill cells, and clinically has shown good responses. So patients are responding to DARA, but unfortunately most patients relapse. So we need to do better with our implementation of DARA. But I want you to keep in mind DARA and its associated receptor CD38. Okay, multiple myeloma is a disseminated disease because it's in the bone marrow. It can potentially be anywhere your bone marrow is. And I wanted to show this slide because it's particularly cool, if that's the right word. This is a first in human study that I'm a part of that we performed at City of Hope, where we tagged daratumumab with copper 64, which is a radioisotope that's imageable on PET imaging. And what you see here is an FDG, which is the clinical standard of PET imaging, which is a metabolic scan, as compared to the copper labeled daratumumab. And what I want you to notice here is that black is in this image on the left is regions of uptake, just like the white and yellow regions are on the right-hand side. And what you should notice is that it's really everywhere. Okay, it's in very many bones in your body are involved, the spine, pelvis, even the arms and legs. And interestingly, there's uptake and activity in the calvareal region of the skull too. So what's cool about this is that, this is really, as a first in human imaging study, one of our first glimpses, besides the Memorial group that is using a zirconium agent, but whatever, our first glimpses of really where multiple myeloma is and how disseminated it is. So this presents a challenge for therapy because you have to get your treatment to all sites of disease. Combination therapies in multiple myeloma always are challenging, and this group I know will appreciate that, because how to determine dose, timing, and sequence of the therapies is there's no real clear algorithmic way to do that. And this combination I'm going to tell you about today, radioimmunotherapy and CAR T-cell therapy, has its own challenges because the RIT has pretty unique radiobiology with it, as well as potential toxicity. And CAR T-cells are a living therapy, and they have their own kinetics and dynamics that you have to take into account. So this is where we come in on the math and modeling and radiation physics side, is to help figure out how really to do this combination therapy in a smart way. So part one, radioimmunotherapy. Radiation therapy, which you might be more familiar with it as a general therapy, is the most common definitive cancer therapy. The idea is that ionizing radiation comes in and damages the cell. It can damage any part of the cell, really, the membrane, the DNA, the mitochondria, anything. But radiation therapy is typically given with we call external beam delivery of photons or X-rays. And in this particular delivery method of radiation therapy, we know where the radiation is going. We know where the dose is. We're delivering it, we're prescribing it, so we know its spatial and temporal distribution within the organ, the target tissue, the target lesion, and then the organs at risk around it. But targeted radionuclide therapy is different, because now what we're doing is delivering radiation through a radionuclide injection into the body. And that radionuclide can be diagnostic, like I showed you with the copper PET imaging, or it can be therapeutic. And so there's this whole field of theranostic agents and theranostic approaches to cancer, which I'm really very interested in. But targeted radionuclides are having a real big surge in interest right now. You may have heard of lutetium, Lutathera, dotatate, somatostatin receptor targeted lutetium for neuroendocrine tumors. There's yttrium-90 for B-cell lymphoma, radium for brain, for bone metastasis, all kinds of applications. This is just a few taken from this reference here. But what we're interested in here is the accumulation of the dose in the tumor, emission of the radiation over the course of the radionuclide decaying, which is different from the external beam delivery, and the pharmacokinetics of the molecule. So because I'm going to be talking a lot about radiation, I just want to clarify a couple of quantities that you may not be familiar with. There's two kinds of radionuclide mechanisms that we're going to talk about. One is a beta mechanism, a beta decay, and then the other is an alpha decay. A beta decay is where the radionuclide, which is, remember, is a radioisotope. So lutetium's atomic number is usually 71. It's overloaded to have 177 neutrons in it, and when it decays, because it's unstable, it emits an electron. That's in contrast to let's say actinium with an atomic number of 89, which is overloaded to 225, which emits a hydrogen nucleus, basically a pair of protons and neutrons as the alpha particle. So alpha and beta radionuclides are what we're going to talk about today. And so one quantity is activity, which is how many disintegrations per second are delivered. So the unit there is becquerel. So one disintegration in 1 second is 1 becquerel, and it has an equivalent unit of curies. 1 curie is 3.7 times 10 to the 10th becquerels. And then energy, megaelectron volts in terms of joules, and then absorbed dose is just the energy per mass delivered. So I'll talk about becquerels and gray, absorbed dose, gray, alpha and beta emissions. So what is a radioimmunotherapy? Radioimmunotherapy is targeted radionuclide therapy delivered with antibodies. So what you have pictured here is an actinium-225 radionuclide attached to an antibody, and over here is a lutetium, a cartoon of a lutetium and a tagged antibody. And there's really been a renewed interest recently in alpha emitters in radioimmunotherapy, and I could give a whole talk on that. I'm really, really interested in that, but I'm just going to say at a very high level that alpha particles and beta particles are different in their path length and their energy disposition. So alpha particles, which is the actinium-225, have a very short path length on the order of microns, tens of microns, and deliver a lot of energy. These dots are where the energy is deposited and potentially damaging cells, as opposed to beta particles, which have much longer path lengths, much more sparse energy deposition. And where the energy is deposited over a path length is called linear energy transfer, LET. So alpha particles are high LET, beta particles are low LET. Alpha particles have short length, beta particles have long path length, okay? And we're going to talk about both of them. So when we think about mathematical modeling of radioimmunotherapy, hopefully I've set up well enough to what the quantities of interest and the challenges are. What we want to do, what we want the model to capture is the effect of the radiation on the cells, so the cell kill. We also want to capture the decay of the radionuclide, because the radionuclide's decaying with either alpha or beta particles over time. We also want to, of course, model the proliferation of the tumor cells and the clearance of dead cells from the system. And I'll motivate each one of these topics for you. But the first two I want to show is the effect of cell kill of the radiation, the decay of the radionuclide. So this is where the model formalism we take is a hazard function kind of formalism, where you look at the rate of change of the surviving fraction of cells being just proportional to the number of surviving fraction of cells available. A very simple kind of formalism, but it's related to the good old linear quadratic model. So I know Heiko said he couldn't be here today, and I don't want to get into any kind of big debate over the linear quadratic, but let's just say that linear quadratic is a dose response model in radiation therapy. It relates the number of cells that survive after a fraction dose of radiation given in gray by either linear or a quadratic component. And the lutetium, as being a beta emitter, has both linear and quadratic components, and the actinium alpha emitter has only the linear component, very high LET, very straight dose response term. But remember, what we want to do is not just have a surviving fraction function, but the radionuclides decaying over time. So a hazard function takes actually kind of an expanded form from the simple linear quadratic. It's still linear and quadratic, but it has a decay rate of the radionuclide. So the parameters of this model are the alpha, which is the linear parameter, linear and quadratic, beta, which is the quadratic, R0, which is the initial dose rate, a lambda p, which is the radionuclide decay constant, and gamma, which is the cell repair rate constant. And what I want you to notice here is that there are 1, 2, 3, 4, 5 parameters in this model, but 2 of them are measured and 1 is fixed. So the R naught is actually measured. It's a known quantity, which is the injected activity. The radionuclide decay constant also is known just from the properties of the radionuclide. And the ratio alpha to beta is fixed for reasons that we could debate if anybody's really interested. So that really only leaves 2 free parameters, alpha and gamma, in that model. Now, how do we expand that to include the proliferation of tumor cells and clearance of cells from the system? As you think about a 2-compartment model where we have non-irradiated tumor cells given by VT, which proliferated at a rate rho. They're exposed to radiation and become irradiated at a rate KRX. And then once they're irradiated, denoted by VR, then they're cleared from the system at a rate KCL. And that gives us a very simple, as far as our mathematical modeling world community is concerned, 2-compartment linear model where we have an equation for VT, which is rho VT minus kappa RX or KRX times VT. And then this KRX, because of this hazard ratio formalism, is given by that expanded and called dose protraction factor, which accounts for the decay of the radionuclide. And then the exposed population are exposed at a rate KRX and then die and clear at a rate KCL. And so if you think about this full model now, we really just have 3 parameters, rho, which is the tumor cell proliferation rate, KRX, and KCL. And it turns out that we can measure directly rho, so that leaves us in some aggregate sense kind of with only 2 free parameters in this model. Because this audience might be interested in how this looks like, how this works, how the dynamics of this model are affected by changing the parameters. We did here's just an illustration. So A, B, C, and D here on the right are showing what happens to the predicted tumor growth curves when you vary one parameter in isolation. So if you vary the injected dose, the activity from 1.5, 1.8 kilobacterels up to 7.4 kilobacterels, you just see a delay in the tumor growth curve. Same if you change very similarly anyway, if you change the alpha, which is the radiation response coefficient, the linear part of the linear quadratic model. Rho, the tumor proliferation rate, and the one that's of most interest here, I think, or the one that's least intuitive, is the clearance rate. And what you see when you vary the clearance rate out of this model is a big change in how the tumor growth dynamics are happening during therapy, but convergence of all those dynamics after therapy is over. So we're going to really think about how, when we fit this model to data, what the interpretation of these different parameters are on the effect.
Before you move on, Russ, let me ask you a quick question. So that classic kind of alpha beta model, one of the kind of critiques that people often leverage at it is that you can't really get good measures of alpha and beta for specific tissues. And they potentially are different in specific tissues. So is that something that you've quantified specifically in myeloma?
So I'm going to show you how we do biodistribution data and how we look at specific different tissues. But that's a really good point, Sandy. And we haven't done that analysis yet, but we have the data to do that, actually. One of the next slides is the biodistribution data. So the experimental design that we did here, mice are injected with multiple myeloma cells and then 1S myeloma cells. After a period of time, they're given either actinium or lutetium radioimmunotherapy. They're followed serially with bioluminescence imaging, and at the endpoint, we do a PET scan to do the biodistribution. And in case you haven't seen bioluminescence imaging before, this is kind of what it looks like. It's photon flux through luciferase gene reporter system. And so here's what the experiment looked like. So in the lutetium cohort, we have 4 doses of activity in microcuries, 50, 100, 200, 300. And then actinium, we have 5 doses, 25 up to 600 nanocuries. And you notice the order of magnitude difference in the unit of injected activity. And there's a reason for that, which I'll show you. But the controls are untreated mice, from which we estimate the proliferation rate. Daratumumab, the antibody alone, so we can see the action of the antibody without the RIT, and then the RIT tagged with a non-specific antibody, so trastuzumab, which is a monoclonal antibody against HER2 for breast tumors. And so that shows us what the action of the RIT is without its specificity to CD38 cells. So here's exactly the next slide that Sandy was asking about. Here's the biodistribution data that we look at. So at the endpoint, the mice are imaged with, again, with this copper labeled PET scan, and we can look at each organ of interest and relate the activity, injected activity, into an absorbed dose. And so we don't actually use this to measure tissue-specific alphas and betas, but we could, and it's a very good idea. So I'm going to write that down, but that's how that looks like. And then here's how the untreated data looks like to measure the tumor proliferation rate. So here's the growth curves of 5 of these mice, and you can see that there's very little variation, very good reproducible model. So if you just fit an exponential curve to those lines, you see a very small variation in the distribution of proliferation rates from 0.32 to 0.42, which translates to like a difference of a half a day in doubling time. So anyway, that rho then becomes a measured quantity, I'll say, rather than fit to data. Of course, it is fit to data, but anyways, it's derived from an experiment that's specifically used to measure that parameter. And here's what it looks like when we fit the model to the data. So here are the 4 dose levels in the lutetium cohort. The squares are the data and the lines are the model fits. And so you see the model fits the data very well, only with 2 free parameters. And what I also want you to notice is that is the x-axis. So the mice are living out to about 40 days here in the lutetium cohort. In the actinium cohort with 5 different dose levels, you see the axis going out much farther to 60 days, 70 days. And the units there being nanocuries as opposed to microcuries basically are just showing you here that the model fits the data really very well. And so now what we can do is use the inference from the parameters to understand what the differences are of these different doses in these 2 RITs. So here's what this looks like. So here's alpha, the top row, alpha, the radioimmunotherapy response parameter given for actinium on the left column and lutetium on the right column. And what I want you to notice here is that there's a nonlinearity in injected activity. It's very high for low activity, 25 nanocuries, dips down for 50, comes back up again for 100, and then goes back down again to all the way to 600. And you think, okay, well, why is that? Well, it turns out that 600 is overkilling. Okay, 600 is actually toxic, and it results in overkilling of the myeloma cells. So they're radiosensitive to the point that they are completely ablated, but it's also a toxic level. Why is the 25 nanocurie level higher than the 100? Well, this is interesting in another sense, and it took us a while to think about this, and that is because the 25 nanocurie level has effectively no effect on inhibiting the tumor growth. And so the cells are at once extremely radiosensitive, but also over proliferating and overcoming the effect of the radiotherapy. So we use this to identify 100 nanocuries as a potentially ideal. I don't want to say the word optimized in like this kind of community, right? And I wouldn't anyway. I'm not saying we optimize the dose, but certainly we want to look at 100 nanocuries more in the actinium. And then what I want you to notice in the lutetium model fits is very little variation across doses. So you don't see a lot of change in the radiation response parameter, and you look at the 2 curves. Although the lutetium flattens out the growth curve quite a bit, the mice still die, and they die very early, as early as 25 days. Some of them make it out to 40, whereas at the actinium, they make it out to 70, or in some cases almost 80 days. So the mathematical model really allowed us to quantify the effect of the radioimmunotherapy over time, and to compare 2 different radionuclides, the alpha emitter actinium and the beta emitter lutetium, and to identify a potential target dose for a subsequent study, which we actually use. And that's what I'll show you in part 4 here, is that we're going to use that 100 nanocurie dose based partly on this analysis for the combination therapy. Okay, part 2, so CAR T cells. I think I think people are broadly familiar with CAR T cells, so I'll just give a very quick introduction. Chimeric antigen receptor T cells, an engineered T cell, which takes the specificity of antibody with the action of a T cell and merges them together. So the T cell now has antibody fragment associated with it to give it sensitivity to whatever that antibody is specific to. And the way this works is that the patient undergoes apheresis or collection of T cells. The T cells are expanded and activated, and then their lentivirally transduced gene construct is inserted into the T cells so that they express this particular construct, the chimeric antigen receptor. They're expanded and then they're placed back into the patient. And there are several CAR targets for multiple myeloma. If there's any multiple myeloma experts on this call, I hope I didn't leave out your favorite one, but there's CS1, BCMA, SLAM-F7, GPR, CD5, whatever. There's several different targets of interest that are being looked at right now in CAR T cell therapy. The modeling I'm going to show you is not specific to the target, it's just the general action of the CAR. So I wanted to have a brief comic relief here before I start going and pretending, giving you the impression that I know what I'm talking about with immunology, because I don't. It's really complicated. This article I saw on Twitter, our favorite platform, from The Atlantic, published a couple of days ago, Immunology is where intuition goes to die. I love this article because it starts off with a joke, and so immediately you get a good sense from it. So here, I'm going to tell you the joke. An immunologist and a cardiologist were kidnapped. The kidnappers threatened to shoot one of them, but promised to spare whoever's made the greater contribution to humanity. The cardiologist says, well, I've identified drugs that have saved the lives of millions of people. Impressed, the kidnappers turn to the immunologist. What have you done, they ask. The immunologist says, the thing is, the immune system is very complicated. The cardiologist says, just shoot me now. Okay. I hope you find that funny, because I certainly do. Just about every immunologist I talk to has said, the thing is, the immune system is very complicated in almost every conversation we have with them. So it's very complicated, but let me just give you a highly simplified mathematician's view of some of the stuff that we're interested in this particular context. Okay. For the purposes of modeling CAR T cell action in a very controlled, limited environment, we think modeled with a predator-prey like equation, predator-prey like model, which we recently published in Journal of Royal Society Interface. This is a very simple model that has a logistic growth in the tumor cell population denoted V, and then a mass action like killing between the tumor cells and the CAR T cells at a rate kappa 1. And here's the important point of this model. When the mass action law, the rate constant governing the mass action law in the CAR T cell equation, dCdt, kappa 2, can be positive or negative. So we don't enforce a sign on that rate constant. And the CAR T cells can die at some rate theta. But this rate kappa 2 has a complicated interpretation that I'll show you. It can either reflect the proliferation of CAR T cells, or their exhaustion, or their death, depending on what the value is relative to the tumor cell proliferation rate. And we used this rate constant kappa 2 fit to data to really make some, I think, interesting inferences about how CAR T cell dynamics can work or not. Anybody who's taken a mathematical biology class and has looked at one of these equations, because it's in Lee Edelstein Cachet's textbook, it's in Jim Murray's textbook, will know that it's structurally unstable in some sense. It exhibits some bifurcation in the parameter space, and I'll show you how we interpret that here. If the ratio of the T cell death rate theta and the tumor cell proliferation rate rho is very close to 0, or exactly 0, but close to 0, and the product of kappa 2 and the carrying capacity K over the proliferation rate rho is positive, then you have a successful CAR T cell treatment. And this is a phase space diagram showing the cancer cells versus the CAR T cells. And the important part of this diagram is that it predicts that even in the case of successful CAR T cell treatment, where the cancer cells are eliminated as a stable state of the system, there's the possibility for a transient progression. And this is a very well-known phenomena in immunotherapy and in CAR T cell therapy in general, this concept of a pseudo progression, that the cancer or aspects of the cancer microenvironment, inflammation, or the tumor size itself, even maybe, can temporarily progress. It can grow before it eventually responds. And so this is an important prediction of this model, is that there's a transient progression before eventual rapid response in this parameter regime. In contrast to when CAR treatment fails, you can have the possibility of a transient response, a transient regression before eventual progression. And being able to differentiate these two potential situations is very, very important. And I'll show you why, how we use it with data. The third case is a little bit less interesting to us because it seems a little bit less physiologic. We don't have really an opportunity to observe it, and that is this typical predator-prey-like dynamic in which both populations oscillate and they converge on a stable equilibrium in which both populations coexist. The nature of cancer therapy, particularly multiple myeloma therapy, doesn't allow us enough time to observe this kind of dynamic. And so there is a fundamental ambiguity, let's say, possibly when you have a borderline case like that with the parameters, which is something that I think only for this group I'd take a minute to talk about. And that is that, you know, when you have a mathematical model, one advantage of it being simple is that you can start to understand it. And you can understand tricky cases like this, in which the system may exhibit a bifurcation. You may be fitting your model and getting parameter estimates, and you may be in a situation where the dynamics are could change, and if the model's too complicated, you may not be able to appreciate that you're on the border of a bifurcation in the parameter space. Anyway.
Yeah, I appreciate that, Russ. But I mean, a caveat to that is the model could just be wrong.
Which one? The simple one?
Yeah,
That's the caveat.
Yeah, that's true. Yeah. Okay, so we have a very cool experimental system to look at this. If you haven't heard of Excelligence before, this is extremely cool. It's an in vitro model, so also talking about simplifications. This is extreme simplification, but it gives you something particularly cool. This is a model system where cells are adherent to the bottom of a plate that has what am I trying to say? Electrical terminals, positive, negative terminals that measure electron flow or electrical impedance through the dish. And when there's cells, the impedance is inhibited and you get a signal. And it's very, very strongly correlated with the number of cells in this signal. And so what we do is some experiment where we plate cells in the dish, they attach and spread to the bottom of the dish over about a 24-hour period. And then we put CAR T cells in. And after the CAR T cells are in, then you get to watch the dynamics play out. This red curve is with no CAR T cells, so you see the signal just increase, so proliferation until the cells become confluent or reach their carrying capacity. And this blue curve is with adding CAR T cells, and you see the signal die out, which corresponds to the cells dying. And what's cool about this is that this picture here, this graph, is the data. So the data is taken, measurements taken every 15 minutes. So basically continuous data, okay? There's, from a modeling perspective, it doesn't get much cooler than that to have continuous data. And one thing from the modeling perspective I just wanted to show you is that when you look at this curve, this growth curve, you might notice that it's concave down. And again, this is something I would only just make a point of for kind of an audience like this, is that almost every growth law that I'm familiar with starts as concave up. Exponential, logistic, Gompertz, anything, right? Starts as concave up. You have to think about what kind of growth law would produce a concave down initial growth curve. And it turns out it's a spatial phenomenon. I'm very familiar with this particular equation, Fisher KPP equation. And it's the result of the cells adhering to the bottom of the plate and spreading out and diffusing. And so it's extremely cool to show you that the data here in part C and the fit of the PDE model to it is like about as good as it gets in terms of model data fitting. So that was just kind of very cool to see that this is a good representation of what's going on there. But we don't model the data while the cells are attaching.
Russ,
we're just
can I ask a question? Sorry.
Yeah.
Why is there any reason why it fails near the end? Is it hitting the end of the dish?
Yes, it's hitting the end of the dish. Yeah.
Okay. Thanks.
Yep. Yeah, exactly. Yeah. I know this is what I asked Prativa too. I was like, but what about these last points at the end? It's like not enough to fit, 98% of the points.
No, it looks great though. Yeah.
Yeah. Okay, so then what happens when we fit this model to different tumor cell lines? Okay, so PBTs are patient-derived brain tumor lines. Two of them, PBT 138, PBT 030, and then HT 1080 is a immortalized fibrosarcoma line. And what you see here is, I mean, again, pretty remarkable fits to the data, and that's cool. It's just fun and nice to see as a modeler, right? So the untreated curves and their fits are in red, and then the treated data and the fits are in green here for each of these lines. And then the trajectories are plotted in the phase space, and that's what is also interesting to infer about this model is that the Excelligence platform doesn't measure the CAR T-cell abundance. CAR T-cells aren't adherent to the bottom of the plate, and they're very light and don't really register anyway. And so we're able to use the model to actually infer the dynamics of the CAR T-cells without any direct measurements of them. So that's cool. But the model correctly predicts a transient progression before eventual regression in the two responsive lines, 138 and 030. And then, because it's fit, and then also the HT 1080 line, which is unresponsive, is in that regime where it quickly progresses. If you look at the model parameters across cell lines and then across CAR T-cell doses, you notice make a couple interesting observations. One is that the CAR T-cell killing rate, which is the great constant kappa 1, decreases with increasing CAR T-cell dose, and you have to think a little while for why that might be the case. And it's very consistent. You see across triplicates and across cell lines and replicates, is that the more CAR T cells you have in the dish, the less each individual CAR T cell has to kill. If you have very few CAR T cells in the dish, they become activated and they do their job very quickly, and they have a very high killing rate. And so it's kind of like a crowding effect, and very not necessarily intuitive result, I would say. And same with the CAR T-cell proliferation or exhaustion rate constant, kappa 2. So that increases with increasing CAR T-cell dose. And that also is interesting to say that the CAR T-cells are changing their behavior based on how many there are in the environment, how many of them are working together. And the CAR T-cell death rate is pretty much flat and constant across all cell lines and doses, and that's good. As a good, kind of stable parameter. We looked at how these rate constants depended on the level of antigen expression. So there are two lines, the top row and the bottom row are two separate lines, red and green, the HT 1080 line and the PBT 138 line. And what you see is they're colored from light to dark in terms of from low to high antigen expression. And what you see here is that the killing rate constant and the exhaustion rate or proliferation exhaustion rate are non-linearly related to the percent of antigen positive cells in the dish. And this suggests, although we're following this up, right? We didn't have a conclusive answer in this particular paper. We're following this up, but it suggests that there's an optimal efficiency or optimal killing rate of the CAR T cells as it relates to the number of antigen positive cells. And I'm just going to leave that at that. If anybody wants to talk about that, they can ask.
Russ,
Russ,
yeah. Joel, can I dive in with a question? This is like so awesome.
Hey, Joel.
Yeah, hey, Russ, great to see you. And I love predator-prey models. So this is assuming, or not assuming, you are now predicting, at least in those first two ones, that the T cells themselves are proliferating without any feedback through other elements of the immune system. Is that
remember? I do have to stop you there, Joel, to say that there is no immune system, so it's just in the dish.
That's right. So you would expect just in the dish, this is just being naive, that the T cells themselves would not be proliferating that much, or is that wrong? Because you're predicting then that if your kappa 2 is positive, these guys are actually proliferating like a predator, and that defies my understanding of the immune system.
Yeah, so this is something we talked a lot about, and there is lots of evidence, both published and anecdotal and local, to suggest that when T cells encounter a target, they can be stimulated to proliferate or
They can be
to die. Yep. So an interaction of a T cell and a target cell can result in the T cell increasing its proliferation rate. Yeah, absolutely.
Russ, thank you. That's fantastic.
Yeah, and it's a very unintuitive kind of dynamic, very important one. Yeah. Yeah, thanks, Joel. So then you ask the question, okay, well, how do the parameters relate to the tumor growth rate? And you would think that the CAR T cell killing rate would correlate with the tumor proliferation rate. And in fact, it doesn't at all. What you see down here, kappa 1, doesn't correlate at all with the tumor proliferation rate. What the correlation that we find is between kappa 2 and rho. So how the CAR T cells are proliferating or exhausting as it relates to the tumor cell proliferation rate. And so what it means is that if the tumor cell is proliferating very fast, the CAR T cells become exhausted. And that makes intuitive sense because the CAR T cells, now how we define exhaustion, hopefully Kim's on the line and she'll ask me, how do we define exhaustion? This is effectively the only question we got in the review of this paper. And defining T cell exhaustion is very difficult from a biological perspective. We have only here a mathematical definition of it, and that's the best I can give you right now, but I'm happy to talk a lot about what we think about that in the future. But let's just say that from a mathematical standpoint, the T cells become exhausted when the tumor cell is out proliferating the T cells. Okay, part 3, the combination RIT and CAR T cell therapy. So we just literally smash those two models together that I showed you, the radioimmunotherapy model and the CAR T model, and ask questions like, which should we give first and in what dose? And what I want you to notice here, this again is very preliminary work, is that you can get a very similar outcome if you give a low CAR T cell dose followed by a high radioimmunotherapy dose as you do if you give a high CAR T and then a low RIT. And that definitely was not intuitive to our collaborators, let's say, at all. So the question now is, okay, well, which one do we prioritize? How do we dose, time, and sequence them, right? Those are the motivating questions I gave you at the beginning. And this is how. So remember that what we learned from the previous experiments, one is that the actinium was better than the lutetium in terms of its radiotherapy radiation response parameter alpha, also in terms of how many cells were killed, in terms of how long the mice lived. That 100 nanocurie dose was in some sense better than the others, and that a high CAR T cell dose should be, okay, so based on those considerations and the fact that the actinium, the alpha emitter radioimmunotherapy can have bone marrow toxicity, we decided to go with a high CAR T cell dose followed by a low RIT dose, because low 100 was not too low, but not too high, kind of in that sweet spot, right? And so what we did to test that was we did an experiment.
Russ, before you go on, isn't there some discussion about radiotherapy being potentially immune stimulatory? So could there be some logic to giving that before as opposed to after?
Yep.
And potentially, could it damage the CAR by giving it after?
You're absolutely right on both counts, Sandy. So I'm actually going to show you the mechanism of resistance idea, which relates to one of those ideas, one of those points. And then also, you're right. Part of the reason we did the RIT after the CAR T was because we didn't want the RIT to kill the CAR T, any residual RIT. But like I said, this is work in progress. So there's a lot more questions to ask here, because you may be able to have enough control with the RIT and wait for the RIT to actually decay out and wash out, and then give the CAR without having to influence the CAR, and it may actually stimulate the behavior of the CAR. So that's
the timing becomes critical, right? But then also how you model, how you integrate the two models becomes critical.
Right.
So how are you doing that?
In what sense? The math sense or the experimental sense?
In the math sense, how are you adding the kind of predator prey with your alpha beta radiotherapy model?
Yeah, so I have it written right here, just in terms of heavy side function. So we literally just smashed the two models together.
Yeah, but that's not going to necessarily, provide synergies or anything like that, right?
No, that's right. So yes, that's right. So there's lots more work to be done here, right? So this is work in progress. Yeah, and in fact, this work is only about 6 months old and not published yet. So I'm going to show you the results of one experiment that we're in the course of repeating right now.
Okay.
So, but yeah, good point. And this is the kind of thing I'd love to talk about with this group, because I know you guys all think a lot about this stuff. So the experiment that we did was to, very similar to before, we have mice that are injected with MM1S multiple myeloma cells. They're given a dose of CAR T like you see here, and a time later, then they're given the actinium, radioimmunotherapy, and then the biodistribution at the end. They're followed with imaging, and that's how we measure tumor burden over time, the bioluminescence imaging. And here's what that data looks like. So we have untreated, we have CAR alone, we have RIT alone, we have a mock CAR with the RIT, the CAR and the RIT, and then a CAR and an untargeted RIT in terms of controls. And the mice tolerated all combinations. Our measure of toxicity was just whole body weight. We also have bone marrow toxicity biodistributions at the endpoint that we're not done analyzing yet, but we do look at that. And then in terms of survival, the combination just did a lot better than any other, certainly than the monotherapy. And then it was the combination with the targeted RIT and the targeted CAR did better. The mock CAR also had some activity in this model, which we suspect is donor specific, and there's a whole story to be told there. But anyway, the combination did a lot better, and so that was promising. So we're repeating that experiment. Okay. Last topic, not too long. I know we're getting close to the end here, but what about resistance? And this is where I would really like a lot of feedback and discussion here. So what about resistance? Okay, so is DARA therapy selecting CD38 negative clones? And this is my collaborator, Flavia. She likes to think about it in terms of a tree rather than kind of a clonal architecture diagram. But anyway, is it selecting off the negative cells? Well, it turns out it's not really. So if you look at both the surface expression of CD38 on the multiple myeloma cells in the bone marrow, and also look at the gene expression, it doesn't change if you compare patients that are refractory to DARA versus patients that are refractory to other therapies. So the CD38 fraction of cells isn't actually going down. I mean, it is a little bit, so it's not as if it's exactly 100%. Sure, there are some samples down here that are decreased, but I mean, in terms of these human samples that we looked at, there really wasn't as big a change as you might expect. So then the next question is, what is it affecting? Is the DARA affecting CD38 on other immune subsets other than the multiple myeloma plasma cells? And is it mediating multiple myeloma cell killing in some other way? And that's where I think is the interesting idea here. So CD38 is changed, is downregulated in non-plasma cells in the bone marrow, and in monocytes, and in NK cells. So it's other immune subsets that are responding more to the DARA than the multiple myeloma or plasma cell populations are. And this is just another way of showing that. That's just, I mean, a little complicated, but to say that it also induces NK cell activation, the presence of DARA does in these patients. And this was published very recently in the journal Leukemia by our group. Of course, not me, but people, our group as the royal we, right? So based on these preliminary data, we hypothesize that DARA-acquired resistance is actually not caused by selection of CD38 negative multiple myeloma cells, but it's actually driven by phenotypical changes in immune effector cells. And that this will lead to a characterization of immune changes over time, which we can start to model and just dissect the actual immune-mediated mechanisms of DARA-acquired resistance. And here it's important to make the note that I'm talking about DARA as an antibody monotherapy. And this is why I set up at the very beginning why we can do better with DARA, because the RIT is attached to the DARA. And the combination of CAR T with a RIT-mediated DARA may all improve, may address this question very kind of, in we think a novel way. So we are investigating this hypothesis with a prospective clinical study. So we're following patients prospectively that have received DARA and any other immune modulating drug with peripheral blood and bone marrow at a baseline sample before DARA, and then at these time points after until progression. And we're looking at the cells through CyTOF, which is like a high dimensional flow cytometry kind of measure, single cell RNA sequencing and single cell T cell receptor sequencing on all these samples too. Very, very big effort and big study to look at all this. So we don't have any answers yet. Again, this is a very, very new unpublished work that's ongoing. And this slide will be unsatisfying for all the math people there because it's very vague. But the point is that the model that we're building, the model that we're thinking about is really tracking the immune suppressive populations, the immune activating populations through these the CyTOF and sequencing and flow cytometry and all kinds of other ways of measuring cell populations, peripheral blood, and bone marrow over time, with the hypothesis that it's in fact this dynamic that's driving the response or not to DARA. This divergence between the immune suppressive cells and immune activating cells. Okay, so summary. From my title, I put this title in there, like I said, because I wanted to kind of illustrate what I think is one of my career goals and why I came to City of Hope is to really integrate the bench and the math and the bedside all together. So from the bench, we have local to City of Hope, local to our investigators, radionuclide, radioimmunotherapy development and CAR T-cell model development and manufacturing on premises. The math contribution was in terms of doing radiation physics, doing dosimetry, which is important, but not all that exciting, let's say. But then modeling the action of the radioimmunotherapy, modeling the action of the CAR T cells and their dynamics, and ultimately trying to work those together into getting a combined therapeutic approach. And then on the bedside, we have our first in human PET imaging to understand the dissemination of multiple myeloma cells in humans, starting to get to both CAR T cell CAR T cell implementation in the clinic, but also potentially CAR T cell imaging. I didn't really talk about that here, but we can do that. And then prospective study of DARA resistance. And then when we say back again in the title, the idea is that we're taking all the things that we learned in the bench and the bedside and with the math to try to design a phase 2 study with the combination therapy. And ultimately we want to try to, like everyone, figure out how to overcome immune mediated resistance and how to improve combination treatment schedule. Okay. And so this is the last slide to say the advertisement that Philip and I and this really, really friendly looking guy, Victor, he's actually a nice guy, and Lizette are putting together a special issue in the journal Cancers on mathematical modeling of cellular immunotherapies in cancer. Jeffrey just put it in the newsletter that came out today, so you can see more information about it there. And thank you again for the invitation. Thank you for all the people that I collaborate with. I think the coolest part of my job is getting to work with all these really smart people in all these different areas and working together to try to do something important. And I'm very lucky to have collaborators that recognize and see that mathematics and mathematical modeling can be useful, and often ask me to be involved in things that I might not otherwise have been involved in. So thank you.
Nice. Great presentation, Russ. There are bits at the end that I think I'd like you to sort of elaborate a bit more about the mechanisms of resistance. So Is the idea then that the treatment is not selecting for a phenotypic resistance, but for plasticity? Like the cells that actually stay at the end, they're just more plastic and able to switch back and forth?
The myeloma cells
Yeah.
will switch back and forth between what?
Well, you said that there was, and you could go back to the last slide where you talk about the mechanism of resistance. Yeah, CD38. You said that DARA doesn't target those. So
So DARA targets CD38. That's what's trying to be shown here, is the fact that CD38 doesn't necessarily change its expression after CD38 targeted therapy.
Mm-hmm. Okay. So I guess.
So So are the CD38 positive multiple myeloma cells undergoing some kind of phenotypic transition? Like what?
So the way I think of a resistance in most of the models I work with is very, very simplistic. So I don't want to say anything, but I think that cancer is quite complicated. So shoot me now.
Yeah.
So what I'm trying to say is, obviously it's not a question of like cells are either resistant or not resistant. It's just a very convenient approximation to a reality. So when you apply treatment in our models, if you're resistant, then you obviously don't pay much of a price, but if you're sensitive, you pay a lot more of a price. Reality, though, is in many cases there might be some plasticity. So cells can switch some certain pathways that are targeted by those treatments, and some of them are actually more likely to switch to alternative pathways and survive treatment. And those changes are not permanent, they are plastic, in the sense that if you cease to apply treatment in much shorter timescales, those same cells might be able to go back to the original pathway and proliferate potentially at the same rate or at the same, using the same mechanism they had before. Sometimes they don't. But what I'm trying to say is those are not intrinsic properties of the cells, so that when you apply a treatment, those cells that have a particular feature are not necessarily targeted.
Yeah.
So I don't know if the direction of my question is now more clear. Are you saying, with your point, that the mechanism of resistance is such that the cells are more plastic and thus able to sort of avoid the full onslaught of the treatment because this can switch to something else?
No, the point that I and we, again, it's the royal we, so don't make this as being my own personal point I'm pushing too hard. Like I said, some of this is shown in the leukemia paper from Flavia and the other members of the myeloma group, is that the argument here is that it's actually modulating other components of the immune system that result in treatment failure. Cool. So it's that the NK cells are being activated and immune suppressive cells are being stimulated. Mm-hmm. And that it's the combined action of a dysfunction of the immune system that allows the treatment to fail and the disease to progress, and not the specific action of the antibody against CD38 positive myeloma cells.
So it's a lot more complicated. That's great.
I mean, yeah, exactly. A lot more complicated.
But it's very cool.
Yeah. But the idea is that it's actually de-emphasizing the action on the tumor cells themselves.
Mm-hmm. Yes.
Okay. Can I ask a question?
Yes, you can.
Please.
Great talk. I also have some questions about the first part with modeling the impact of radiation. So the purpose is eventually translating into patients. And this animal model, you essentially have very explosive exponential growth upon the transplantation. So you're in the model is just like one proliferation term. But I would assume that in the patients this disease exists most of the time near carrying capacity, where there must be a balance between proliferation and death. So the first question, would this impact the sort of predictions from this model and utility of the model?
Yeah, that's a really good question. That's a really good question. As you can see here, we don't have a logistic or a sigmoidal growth law there. So you're right, it was just exponential. And in the mice, like you show here, the mice certainly grow exponentially if untreated. How does a more sigmoidal growth law, and where if the patients were closer to a carrying capacity or closer to
and in patients it must, because they develop over so much time. I mean, and these are very aggressive models in mice. It's convenient, you can get results very quickly, but the question is, do you have a relevant dynamics if the point is figure out how to treat the patients in the clinics?
Yeah, that is a very, very good point. You know, as I showed at the beginning, multiple myeloma has a 54% 5-year survival rate. So you're right, it probably on the scale of cancers has a lower overall net proliferation rate than others that have a much shorter. I mean, one might hypothesize, right? It's not necessarily true that faster growth rate is worse always, but because it's so disseminated in the body, it could have a lower overall growth rate. In fact, it may be more the opposite. It may be more that it's not close at all to the carrying capacity, but that the very low growth rate would might have an intrinsically different kind of radiation response. That's a very good point. I think one way we could test that is by having, because this is effectively a xenograft model in the mice, it's not a syngeneic model. We could do an experiment where we give lower initial dose or inject fewer multiple myeloma cells, like very, very few, right? And have a much longer onset time and see if starting the radioimmunotherapy at different times had different measurable effects.
But these mice, like they would die very quickly, and like after this explosive growth, they would start looking bad, and they would die if you don't treat them right.
Oh yeah, in fact, this is the only data points we get from them. They only live 30 days. That's it. Yeah. That's the entirety of the untreated growth curve right there.
Yeah, so that's quite different from the patients. And the second question I had
is not necessarily. Well, I would say, hang on one second, just to say that's not necessarily true, because there is kind of principal valometric scaling, right? So it's not necessarily true that's not on the same order of magnitude as what we'd infer a patient proliferation rate to be.
Yeah, I mean, I would assume it's possible to get this data directly by just like examining, markers like either expression and proliferation markers, or doing like Ki-67 on the bone marrow draw or the blood draw. I mean, there are probably fewer proliferating cells in the blood, but it should be possible to estimate the proliferation rates in human patients. It's more difficult to figure out the death rates. I mean, but might not be entirely impossible. But I think it's worth to consider especially when translating into clinical strategies. But another sort of part that was like very curious is that you've shown that there are some treatment groups where mice die despite the disease is under control. So it suggests that the cause of the death is not necessarily this explosive growth of the leukemia that kills the animals. So and one sort of aspect of this, it's targeted therapies, but it goes throughout the bone marrow environment. And obviously we want to kind of kill the tumor cells, and this is where they are, but it must be damaging both the niches and stem cells in the bones. Do you have to take this into consideration? And the question is, would even like the pretreatment of with these antibodies without at the given doses, even without the leukemia, what would be the impact, and what would be the impact if you give this treatment and impact and transplant the disease? Would it behave the same way? Would they proliferate?
Very, very good question. Let me show you something. First of all, I'll say that you're right, that alpha emitting radiotherapy has potential toxicities, and bone marrow toxicities of alphas is actually what took them out of favor, if you will, for a long time, for a couple of decades. They were used, I think I have a citation in one of my previous slides, but anyway, they're seeing a resurgence because there's an idea that we can limit or mitigate the bone marrow toxicity. So you're right, that some of the mice are dying probably not because of multiple myeloma disease burden, but because of RIT-induced toxicity. That's something that we have to keep track of, okay? And that's something we have to watch. What I want to, by the way, this is the toxicity. Now, I wanted to go to this slide and then another one. So this is actually what the tox as measured by animal body weight looks like. And 70% are floor, meaning the mice can drop 30% in total body weight before the IACUC says it's toxic. And I mean, it would be anyway, right? So even very high level doses, the 600 nanocurie dose is still within tox limits, technically speaking, but that's not to say that's not why they died. This is the other figure I wanted to show you that I have as a backup figure, and that is what happens when you give cold antibody prior to the imaging, which might be similar to cold antibody before the therapeutic RIT, right? And this is 0, 10, 45, 95 milligram doses of the antibody that's not labeled with copper 64 to suppress the background signal and increase signal to noise ratio in the PET imaging. And what you see, if you look at the doses, particularly in the bone marrow, is that there really is kind of an optimal, or there's a nonlinear response in the dose, and that you can oversaturate the receptors here at like 95. And so all that to say that part of the way we think about bone marrow protection is, yes, through a cold dose of antibody before the hot RIT dose. Yeah, but you bring up a lot of important points because, you know, and this is what makes all of this really challenging. Multiple myeloma, disseminated disease. Therefore, any treatment you give that's targeted in any manner is going to go to many places. So you have to think about toxicity, you have to think about distribution. And that's why we're trying to marry all these things together, the imaging, the modeling, the preclinical, to try to improve it, because, you know, there really is good evidence with the 5-year survival rate of 54%, there are patients that respond, right? And so we need to figure out why, you know, why those patients are responding, and for the ones that aren't, how we can improve it. Yeah. But distribution of drug and toxicity related to the drug are really important, considerations.
Well, my other point is that, I mean, the impact of the radiation on the bone marrow environment could modify the proliferation and death rate of the disease itself.
Yeah, absolutely. No, that's right. That's absolutely right. And so we have that data. I just didn't show it today because I said we're repeating the experiment that I showed you, the combination therapy experiment. But yeah, we are paying a lot of attention to looking at the bone marrow and looking at the effect of RIT on the bone marrow and on the multiple myeloma cells in the bone marrow and in the niche. I can't honestly say that specifically bone marrow niche is a word that we say very often in this context, but maybe we should.
Yeah, I mean, it's been shown in many studies, and we've seen this as well. So if you introduce the radiation, you induce some senescence in many of the sort of fibroblast type cells within bone marrow, and these start secreting a bunch of cytokines that they don't normally secrete. So your cytokine milieu is now different, and then it must have impact on the composition of the bone marrow in general, stem cell niche, and then proliferation, death of tumor cells themselves. So it's kind of changing the rules of the game with this radiation.
Mm-hmm.
Okay, I've got a couple of questions, but I'm going to let Kim ask hers first.
Oh, good.
Hey.
Hi, Kim.
Hi, great talk. Thank you very much. Okay, so my question's on the CD38 and the, well, first, so when you're showing the slide and you're saying that the multiple myeloma cells after treatment with the CD38 RIT don't downregulate, I believe was the word, the CD38. Is that what we're seeing?
Yeah.
And are we sure it's on the surface still, or?
I mean,
my screen is very big. It's not intracellular. It's okay. I just make sure they weren't hiding it.
Not being a pathologist, I'm going to tell you that it looks like it to me, but that's certainly the claim that we're making. Yeah.
Okay. And so I guess my thought was that you were talking about resistance to the drug is not that they lose their target, of the multiple myeloma cells do, but that you have kind of this exhaustion of the immune cells. Is that accurate? Is what you're saying?
I think that's one way to look at it. I mean, exhaustion is a difficult word to use because it's so poorly defined, but this is basically the idea is that immune suppressive cell subsets increase and immune activating subsets decrease. at a very general level. And that's why I also started with the immunology joke and a reference to you, Kim, is that I'm going to be very careful to not like overplay my hand here, because I'm not going to pretend to be the expert in this area, but that's the general idea.
Okay. So do you think, just by just your current thoughts, I mean, I won't quiz you on this later, but do you feel that the immunosuppression is just happening because you've activated the immune system so much with this monoclonal antibody and all of its ADCC and its complement dependent? Or do you think that the actual drug is really bad for our CD38 happy expressing NK cells, which you depend on for the immune aspect of it? Like, which one do you think it is?
Oh, I think that's a really good question. I think it's probably antibody dependent activation. Yeah, and probably over. Yeah, you're right. I mean, if I was to guess, I would really love to hear what Flavia thinks about this, because this is her pet hypothesis. Really, I can't remember if you got to meet her when you came out, but the idea is, I think, probably more closely related to the fact that the antibody, the DARA, may be overstimulating or overactivating the immune system, and it's a way of like recompensating.
Okay.
That would be my guess. Yeah.
Okay.
That was one of the choices, right?
Yeah. Whether you're just turning on the intrinsic suppression mechanisms, which we would be doing with any immunotherapy. But the CD38 is just so tricky because you're targeting immune cells. So I wasn't sure which direction you thought it might be going in.
Right.
Very cool.
Yeah. Well, yeah, if I was to pick based on what I know, that's what I would say.
Okay. Thanks.
Yeah.
Okay, so since you're on this slide, I'll start with this question. So the immune suppression there, it can be through multiple mechanisms, as sort of Kim hinted at, right? And Locta Volterra isn't probably going to be the right way to try and model that. But in terms of those kind of multiple mechanisms, how are you thinking about implementing that? Or is it still very much fuzzy and you're not quite sure?
It's very much fuzzy and we're not quite sure.
Okay. That's I gave you a good bailout there.
You did, but also it's true. And I said it when I showed this slide, is that it would be unsatisfying for the math people because these functions G, R, and F are just very vague, you know, and so, yeah, this is the study where we're collecting all that data. Like I said, is about 6 months in, 9 months in right now, and has been slowed because of COVID. But you know, we're starting to get the data, and we start to get a better sense of what our idea is.
But anyway, the point was sort of more that, you know, the prey and the predator relationship breaks down when you have, you know, immune suppression mechanisms, right? So, and then my other point kind of related to
but one thing I'll say, Sandy, is that the predator-prey concept really was focused on the CAR Ts. This is something that we hypothesize is the result of the antibody. Okay. So the DARA, yeah. So I don't think I didn't mean to give the impression that the predator-prey like dynamic would be relating to this in particular. This is part 4. So this isn't about the combination therapy. This isn't to do with CARs.
Right, I misunderstood my part. I just thought when you smash those models together, that was just going to be the next logical addition and so on, right? But this is something different.
Yeah.
So, but, you know, coming back to multiple myeloma in general and thinking about how that disease progresses and where it originates, and it talks to Andre's point, which is the bone marrow is a microenvironment, right? It is a context and it has structure. And when you look at those images, it's clear the bulk of it's in the marrow, it's in the bones, right?
Right.
And so there's nothing bone specific about what you're modeling, and that's okay. But I wonder though if there could be some important feature you're missing because of that. You know, you hinted at maybe you potentially would have, say, different radiotherapy responses or whatever that would be more organ specific. And if you think bone versus, I don't know, a different organ, that would be one way to account for it. But I'm thinking about, could there be also differential immune response?
Yeah. I mean, yeah. So I don't have a more specific answer to that than yeah, because we need to think about what scale we're interested in modeling, right? And so here, if you look at the in human uptake, you know, David could probably speak more to this, but you know, not all bones are the same, right? And so the bone marrow microenvironment, the bone marrow niche is going to also be incredibly localized. So I'm not sure if I want to go down the mental path of modeling, you know, whole body niches kind of necessarily, but you know,
but there's ways to do that that are still potentially simple, right? Which is like there's a parameter change for an organ. I mean, it doesn't have to be that you implement the whole gradient of nutrients in a bone marrow resolution. But it's
that's right.
It's just worth thinking about purely because that's where the data seems to be, right? Is there some specific you could leverage there?
Yeah, and one thing that maybe I didn't make very clear is that, you know, part of the strength of this imaging study is that it connects, and this is this kind of theranostic idea, it connects what we can see with the PET imaging to where possibly the distribution of the drug is, right? And so if you look at this image in a person, and then you look at this image here in the mouse, I don't have all the labels down here, but these are all organ specific dosimetries, right? And so you can actually, and this was your first comment, right, is you can actually start to, look at alpha betas or response parameters in specific organs. And maybe that's a way of, you know, getting to a role of the environment without going into too many details.
Exactly.
Yeah.
And it'd be very nice overall, Russ. Thank you.
Yes.
Yeah, no, thank you guys. Yeah. Like I said, I chose this one for lots of reasons because I know that there are people over there that are interested in CAR Ts and are interested in radiation and immunotherapy and in translation and bridging and bridging clinic and biology. And so that's why I chose this one. But, you know, there are other fun things to talk about too that on the more theoretical side that maybe we'll have some other time.
Sure. Thanks, Russ.
Thanks.
Yeah.
I don't think there's any other questions. So
yeah.
Okay.
Have a good weekend.
Yeah, and see you on SMP, I guess.
Yes.
Right.
Bye. Good job.
Bye.
Thank you, Kim.